Welcome to Chapter 1. Let's begin with the basic overview of evolutionary biology and what that's all about, what you're going to be learning about in this class, and we'll get to dive into a little bit of how some of this works. So, evolution is really one of the great scientific changes. It's been a huge revolution in terms of how we think about living things in our environment. And there have been a lot of these throughout the history of science, starting with the heliocentric solar system, Newtonian physics, Einsteinian physics. These represented huge shifts in how we thought about and understood the universe and how things worked. Mendel's pea plants gave us an incredible understanding of genetics that we're still using today. Our understanding of anatomy continues to grow and expand, and we're still doing research on that. In the last 2 years, we've discovered extra muscles and tendons and confirmed their presence, even though they'd been postulated since the 16 or 1700s. Our understanding of DNA is incredibly modern. It's only been since the 1960s that we've really even had that to work with, and look at where we are today. We're having arguments over whether or not transgenic organisms should be allowed, whether or not we should have GMOs, whether or not we should even be able to edit the human genome. Our understanding of evolution is no less revolutionary, and that's really hard for some people to think about in that light. But it really allowed us to start to understand that living things actually change. Once we began to accept this idea, we started to explore why that happens and how that happens. And that's really at the heart of what this class is about, is the how and why do living things actually change. This includes things like MRSA for drug-resistant bacteria. It includes tuberculosis. It includes where Zika came from. It includes how we end up with crossbreeds between Cocker Spaniels and poodles, cockapoos, whatever the heck those are. I don't even know. We've created a lot of strange dog breeds. So living things do change. We see clearly that this does and can happen. Evolution is about how does this happen? Why does this happen? What's driving this? What's kind of strange about it, though, is that this isn't a new idea. In fact, although we're not covering Chapter 2 in this particular material at this time, just very briefly, the ideas for evolution had been floating around. They're not new. These ideas go actually all the way back to the ancient Greeks. When they were looking at rocks, they found fossils in the rocks of fish on mountaintops, and they couldn't explain it except in one of two ways. Either the fish walked up there, or since fish don't have legs, that's kind of implausible, so maybe that area used to be covered by water. It's funny that they didn't really posit an option 3 or propose an option 3, which would have been kind of obvious to me as an old ancient Greek person. Obviously, the gods of Mount Olympus ate fish. But hey, that was them. So people have been mystified by the stuff we've dug up out of the ground and out of the rocks and things that have appeared for a long time. We want to understand what all this is and what's our place in it. Evolution can't answer that part. But it does help us understand how living things change. So what made Darwin's ideas so nifty and so new and such a radical change in our concept of the living world that we're in, he put these ideas together in a way nobody really had before. He saw connections where nobody else had seen connections. And this is one of the key things that makes a really good scientist, is their ability to make connections. He got this new unifying idea that tied together all of these different lines of evidence, all of these things that he'd been seeing, and it went click and it made sense. And that's what made this kind of one of those fundamental shifts in how we understand biology today. There have been tons of studies that continue to support Darwin's idea on evolution and natural selection. Whether you're talking about research that's been done in the past, even before Darwin, to research going on through Darwin's own lifespan, to studies that we're continuing to do today and that we will be doing in the future, all of this stuff still gives us support for these ideas of how things change and trying to understand why they change, what's driving those changes. Darwin actually proposed two major ideas. One was that the wide variety of life that we see came from pre-existing species. Now, this may not sound like a radical concept, but a lot of people's minds, it was, because God created things and things were static and unchanging, and this is just how it was. There's a problem with that. Things do change. Why then would we be able to dig up skeletons of mammoths and things that don't exist anymore? They obviously existed once upon a time. Why do we have some of these crossbreeds of dogs today that we've created? Things aren't static. MRSA didn't exist 100 years ago, or at least nothing like what we have today. What are things going to look like 100 years from now, 1,000 years from now? How will we be different? Will we still have wisdom teeth? Are we still going to make little hairs on our arms that stand up and give us goosebumps when we get scared? There's a lot of questions that we have, so we're trying to understand. Darwin's second major observation is that the fit between a species and its environment is due to natural selection. And that's going to bring up a whole host of other questions. But he thought that it was this process of natural selection that was principally responsible for driving a lot of the change that we can observe and that we can see happening around us. So, what did he mean by natural selection? Usually when I ask students what the first thing that pops into their head when I say natural selection, I get survival of the fittest. And that's great. But I have to ask you another question. What is fit? Is somebody physically fit if they can do 50 push-ups? Is somebody physically fit if they can do 100 push-ups? Where do you draw the line? Do you have to be able to do push-ups to be fit? What if you're not good at push-ups? What if you're just a good marathon runner, but you suck at push-ups? There are a lot of different versions of fit, and not all of it has to do with physical size and strength. This is a major point in consideration. So imagine for a minute that you're a little white lab mouse and you're skittering around in the field at night, because mice are nocturnal, so that's when they come out, and you're hunting for some dinner, I don't know, maybe an acorn or some berries or something, and an owl swoops by. What are you going to do? Run, probably. You're a mouse. Does it matter how big and strong and robust and physically fit of a mouse you are? Probably not, to be honest, because you're not going to fight off that owl. There's no way. You're physically just not strong enough. Can you outrun the owl? Again, they're flying, you're running on the ground. Probably not. It's possible, but it's not super likely. So what are some better options? Not being white? How about being a nice brown mouse or a black mouse, one that blends in with the background that isn't as noticeable at night? Yeah, that sounds like a good plan. It's called camouflage, and we see it throughout the animal kingdom. So this is another example of what can make something fit. It's not necessarily about physical size and strength. It's about being better suited to your environment. It's about being able to do one of two things in your environment that become really, really critical. And that's survive, number one, and reproduce, number two. You do not have to be the best. I am not the best runner, believe me, nowhere close. You don't have to be perfect. You don't have to have even really, really good running skills. You can be marginal, you can even be below average, and sometimes that's good enough. Maybe not in your education, maybe not in your personal beliefs of what you find acceptable in yourself, but as far as nature is concerned, you don't have to have the world's best immune system as long as it's not the worst. You just have to be able to function in your environment. So maybe a better way of thinking of this is survival of the, that works, fit enough. So what we're going to be looking at in this chapter is really a brief introduction to evolution and natural selection. We're going to look at some issues related to that, such as artificial selection, what that is, how that works, and how that can be used as a model for natural selection. We're going to look at antibiotic resistance and where MRSA comes from and how that develops. Conservation biology issues come up even in this first chapter. And even to some extent, we're going to look a little bit at the molecular genetics and evolution in primates. We'll come back to the whole human and ape issue and all of that later on this semester, kind of at the tail end of the semester. And the last thing that we're actually going to look at is the evolution and development of the sex ratio. Have you ever wondered why populations are approximately half male and half female? Is it so we can all find our soulmates? Do you think nature cares about soulmates? Do chickens care about soulmates? What about butterflies? What about dogs and cats? They're not even monogamous, so the concept of soulmate doesn't even work there. So why would they be half male and half female? Interesting question. We'll get to that. So evolution is classed as a science. It is. It's part of biology. And scientists are really like a bunch of 3-year-olds who never grew up. We are obsessed with 2 major questions. How does stuff work, and why does it work that way? We want to know why the sky is blue. We want to know why we find algae forming in these ponds and why they're different and how they're different from those algae over there in those other ponds. We want to know whether life can survive on other planets, or at least I totally do. We want to know what makes us tick at a very fundamental level. Why does our DNA work the way that it does? If we went and found life on other planets, would it have DNA? Would it be recognizable as DNA? Or would it be totally different? So it's all how and why. And we spend a huge amount of time worrying about these 2 major questions in whatever part of biology that is, whether it's medicine or marine biology or invertebrate biology or whatever it is that you may particularly be interested in. You're in this class for some reason. Because it's a scientific field, we have to have evidence. And from this moment forward, you should not take my word or anybody else's word for hardly anything. Always ask yourself, what's the evidence? What's their data? What is their proof? I don't like that word, but I'll use it here. What is their proof that this idea is plausible? Mythbusters. So, a long time ago when I started biology, one of my professors defined evidence as stuff you can count or measure. This sounded like a dumb definition to me back then. Truth be told, it still kind of sounds like a dumb definition. Count and measure. How simple is that? Look, I mastered these skills like back in kindergarten. Woohoo! First grade maybe? I don't know. Counting and measuring? That's not exciting. But the more I've thought about it, the more accurate that becomes. Why? How many fingers am I holding up? The reason I asked you how many fingers I was holding up is because this is not a number that we argue with very much. If I tell you I'm holding up 2 fingers, everybody knows how many I mean. I mean 2 fingers, not 2.5 fingers, not 2 3/4 fingers, 2 fingers, and it's pretty clear. We've got better agreement on numbers. What color is my shirt? Muddy brownish with a water spot. I didn't. Khaki, olivey khaki. I don't know. I'm not good with colors. So there's a lot of differences of opinion on that. People will argue whether it's lilac or lavender. I don't really care, but you kind of get the point. It's a lot easier to take something that we can agree on and I can say, I have a dozen eggs, you automatically know how many I'm working with. If I specify I have a baker's dozen, that means I have 13. So what we're going to look at next is the evidence for evolution. And there are actually a lot of different types and sources of evidence that we have found that all support the ideas that Darwin put forth and our modern understanding of evolution itself. And these are just some of the fields that we actually have lines of evidence from that support evolution, and we're going to work through them one at a time. Starting with fossils. So one of the things that I like about your textbook is that it actually defines what a fossil is. It gives us at least some sort of age estimate on that, and it basically says it's an organism that is 10,000 years old or more, and this is the leftover traces of what it was. Okay. We can dig up fossils, and we can count how often certain types of those appear in the fossil record. We can look, for example, for how many T-Rexes do we find versus how many Velociraptors do we find versus how many, I don't know, pick another dinosaur, your favorite. So we can start comparing these population sizes to see kind of what the balance of things looked like back then. We can measure the sizes of bones and see how big or how small these animals were, how big around those bones were, and what that tells us about how the animals walked and ran. We'll get into some of that in this material throughout this semester. We can track how these availability of bones change through different layers of rock. So if we go really deep in the rock, what do we find there versus what's closer to the surface, like the cat that somebody buried last week. And we can compare these to groups that exist today or groups that we find other places in the fossil record, elsewhere in the world. So we can make comparisons of all of these different types of data, all of this different kind of evidence that we can actually collect. Behavioral data. This is more observational than it is really experimental in many cases, but we can count how often a particular behavior occurs. So we can sit there and we can record how long it takes monkey A to groom monkey B, how long a behavior continues, or how many lice monkey A picks off of monkey B, or whether or not monkey B gets groomed by more partners than monkey A. So there's plenty of things that we can count and measure. This is important because it gives us a way to quantify behaviors or turn it into a numerical form. And then we can take this a step further, and especially for our understanding of evolution, we can actually see whether that offers some sort of advantage. Is there a reason that the monkeys pick lice off each other? Aside from the obvious, ew, that's gross. So does it affect their health? Does it affect their fitness in some sense? Why is this something that happens? We have lots of morphological data. And a morph just refers to a body shape or structure. So what we have here in this particular slide are a bunch of different butterfly wings. They've all got different numbers of spots with different shapes on them. Some of them have big eye spot looking things. You've got the one with sort of the blue stripe through it. You've got variations of all of these different types and patterns and shapes of wings. And if you think about all the other butterflies out there, that's a tremendous variety. So we can actually count how often certain forms occur. How often do we see eye spots versus stripes? We can count the differences in the numbers of types of wings that we see structurally. We can look at bones of organisms, and we can, again, compare these to other species, both living and extinct, to try to get an understanding of what this animal might have been like, especially if it's no longer alive. We do get some data from embryology, and this is actually not the same figure that was in your textbook. I swapped it out. This one's a little bit more detailed, and it actually looks at the development of a mouse all the way from its first half day of life, E0.5 up there in the top left, where all you see is this gray thing. That's the fertilized cell. That's the zygote. As it starts to divide at day 1.5, and it doubles again before day 3, and it keeps doubling and doubling and doubling, and by about 13.5 days after fertilization, you end up with basically a baby mouse. And that's actually kind of neat. So we can actually compare this between how mice develop, how rats develop, how bats develop, small animals. We can even compare this to early stages in human development and see where the similarities are and where the differences are. And this can help us understand certain things about our own biology, like at what point do our tails disappear? Because we are not born with a tail. It just doesn't happen that way. At what point do we actually separate the skin between our fingers so that we have individual fingers instead of webbed fingers and toes? This is another part of development that we go through that a lot of animals don't necessarily. So this leads to understanding of a question that I had all the way back in 7th grade, and I remember this to this day because my teacher had just explained mitosis. It's not that I didn't understand mitosis. That part was pretty clear. One cell splits, divides into 2 identical daughter cells. Yay. But what I didn't understand was the beginnings of this. So starting like what you're seeing in this slide, up there at the very top, on day 0, you've got 1 fertilized cell. It splits, it creates 2 identical daughter cells. Those make copies. Those make copies of the copies. They make copies of the copies of the copies. And yet, by day 13.5, you can see eyes and skin and internal organs and a brain. And so the question I had to ask my teacher is, why aren't we just big blobs? I understood how we could get up to day about 3.5 there on the very top right, but I could not understand how we got to day 13.5. As it turns out, she didn't know either. And I'll come back to that in chapter 13. We will get a much better answer for some of this when we get to chapter 13. That's what that whole chapter is about, is this process of development. but we can totally look at when these structures appear and when they disappear, and how this is similar and different across all mammals, and how this is similar to what happens in amphibians or reptiles, and what the differences are. And to a modern understanding, we can even start incorporating things like what genes are controlling these processes. Where do some birth defects come from? Are there ways we can correct that? And so this is why it's of interest to a lot of different people. This brings us to our molecular data, most of which that you hear about is really related mostly to DNA. We do a lot of work with RNA that's related to gene expression, because that is sort of an intermediate step between DNA and proteins. We'll come back to that in chapter 6. In a modern world, we're actually starting to look at carbs and lipids, fats and oils, to a lesser extent, and how that actually changes things too. For example, your blood groups, whether you have type A blood or type B blood or type O blood, is determined not by a protein exactly, but by a carbohydrate, and what sugars your blood cells actually use to decorate the surface of your blood cells. This is the whole basis for our blood typing, and it's a carbohydrate. That's so cool. So in terms of genetics, we can look at the frequency of genes in populations, how often they show up. What is the percentage of red-haired people versus blonde-haired people, things like that. We can track how long it takes for a particular gene to change. How long has it taken from the time we first started observing for us to show a reduction in our wisdom teeth? Just the other day, watching the Science Channel, they were talking about it in a show, and they asked the question, what has happened to human brain size over the last 5,000 years? Has it increased, decreased, remained the same? As it turns out, it's actually decreased over the last 5,000 years. Our brains are shrinking. How's that for a happy thought? So what does that mean for us? What is evolution? Ultimately, it's all about change, and change over time. This is the process of descent with modification. That part, okay, descent with modification. I'm a modified version of my parents, sort of. Technically not, I'm adopted. But most people are modified versions of their parents. They're not clones, they're somewhere in between. They've got a mix of the genetics from both their mother and their father. So Darwin's thought was that it's this process of descent with minor changes that's responsible for the origin and the maintenance and the diversity of life. This was one of his major insights. Another part of this understanding of evolution, and another definition that you'll commonly see in biology in particular, is that it's the change in the frequency of an allele, a specific form of a gene, how common blue eyes are. And you track this over time in populations. That's specifically the definition for microevolution rather than macroevolution, but it's still true, and that's the definition that I sort of grew up with. So, according to Darwin, for descent with modification, we really need to know the ancestral history of an organism. We need to know who its ancestors were, what those critters were like, in order to really understand what kinds of modifications and changes have occurred over time. That's a little complicated when you deal with the fossil record, because it's not like moose keep written records and family trees. So we do the best we can. And that really is still basically the heart of the process by which we change over time. We inherit certain sets of genes from our parents. Some of those genes we use, others we don't. How that plays out in our biology and then how that interacts with the world around us determines whether we are fit or not fit, whether you become a zombie or whether you escape. What causes that? It gets more complicated. We're actually going to spend a lot of time in chapter 7 and 8 talking about different forces for evolution and how that works in really, really deep detail. For now, in this chapter and the next chapter, we're going to kind of get a surface view of what's going on. So Darwin said that natural selection was the major force underlying modification. Not the only force, but probably the most important, at least in his mind. And today, that's still largely how we look at it. Part of this is driven by genetic mutation. Oh my God, mutants! We're all going to become X-Men! Yay! No, sorry, you're not that lucky. So, genetic mutations are changes in our DNA. You probably already know that part. but these have the potential to change our phenotype, what we look like, what genes we're expressing. These are any observable, measurable characteristics of an organism, including the color of your hair, your physical size, your blood type, even if it's not physically visible because most of us don't show our blood type on the surface. It's still part of our phenotype, who and what we are. Our genetics, our genotype, is hidden. You don't know if I'm homozygous dominant or recessive, or whether I'm a heterozygote for a particular trait. You just know that I have that trait or I don't have that trait. So your genotype is kind of hidden, and in the case of natural selection, a little bit less important. Your fitness, as I've already mentioned, really comes down to one of two things: your ability to survive and your ability to reproduce. And that second part becomes important too, because if you don't pass on your genetics, it didn't really matter if you survived or not. So to be a survivor, to be really, really fit, you have to both survive in your environment and have offspring and pass your genetics on to future generations. The better your genetics do compared to somebody else's, the more fit you are compared to that other person. The same is true for mice in a field. So, over these next few slides, over this next rest of the semester, you should be asking yourself about the evidence that I show for evolution. What is the stuff that we can count and measure? What have we observed? What have we tracked? What do we know? What do we have numbers for? And some of it we don't have good numbers on. It's kind of hard to say, look, we found 3 of these. That's not a convincing number. But we're doing the best we can. So we're going to start with examples of non-natural selection. In particular, selective breeding. Humans have been changing organisms for our own purposes since we began gardening and domesticating animals. Selective breeding is a process where humans decide which plants or animals are allowed to breed, and in many cases even how they breed. Whether this male dog mates with that female dog, or whether it mates with those other female dogs, or what. We've been doing this for a long time. We have bred dogs for specific purposes, horses for specific purposes, crops for specific purposes. So we've modified our environment a lot. It does show us that change is possible, and change is something that is realistic. It's not necessarily to say that it's a perfect model for nature, but it does show us under some circumstances change can happen. Artificial selection is really basically just human-directed selective breeding. So we've bred animals for being the best tasting, the fastest growing, chickens, for example, pigs, the fastest runners in horses, and even some dogs, animals that lay the best eggs, the brightest berries, like raspberries, the cutest puppies. We breed for everything. Some of it doesn't even have to make sense. What is the evolutionary value of being cute? It makes you very likely to be adopted by a human? That would be good. So we're starting with this because artificial selection serves as a really good example for how change over time can happen, and it gives us a model for how natural selection can work. If deer prefer sweet raspberries, they're going to eat the sweet ones and leave behind the bitter ones. So that's where this begins. We've been breeding crops for a really, really long time, and some of these go back 10 to 11,000 years ago if you actually look at the bottom of this set of charts and graphs here. You've got a map that shows different regions of the world, and you've also got this bar graph below it that shows you approximately when these plants first started to be used in both pre-domestication, where they were being wild harvested, and post-domestication, once we started gardening and planting those. You're probably going, 10,000 years, how can we tell if somebody was out picking grasses or whether or not they were gardening? This is just like dumb. Well, we look for clues where human settlements were, for example, and we look for collections of seeds, and that usually tells us something about whether or not these were being harvested and whether they were being raised. So we can actually do some of that. We can compare the genetics today a little bit too and start to get a better handle on whether these show signs of domestication or whether they're still fairly wild plants or animals. So we see the first signs of domestication, some of the earliest stuff going back into Southwest Asia and Mesoamerica. That would be sort of the middle region of America, down there around Mexico. We see development of domesticated crops a little bit later in South America, East Africa, and South Asia. So there's kind of a progression as this sort of spreads out across the world. It's not perfect. You'll notice that there's a lot of disagreement and a lot of different dates in there. It depends on what crop you're looking at. It depends on where you're looking, and there's a lot of variation. Humans aren't consistent. So in the previous question, I asked you what makes a good strawberry to you. You could choose one or more of those options. Most of those are actually pretty good answers. However, how would you breed a better strawberry? It depends on what criteria you chose. If you want your strawberry to be really big, you might want to start breeding for size first. If you want your strawberry to be seedless, you might want to start breeding for that first, although it's kind of hard because seeds are reproductive. That's how they spread their genetics to the next generation. If you want your strawberry to be hypoallergenic so that you don't trigger allergic reactions in people, that's a whole different set of genes you got to worry about. So you have to pick one of those criteria and you have to start doing something. How would you do it? I get a lot of answers to these types of questions, but I'm going to take you through a really simple, very, very easy model. Let's start with taste. I don't care what my food looks like. If it tastes good, I'll still eat it even if it's weird looking. So the color of the strawberry, the size of the strawberry, those are all secondary to me compared to how it tastes. So I want to breed my perfect, most sweet, delicious, juicy, burst in your mouth, wonderful summer ripe strawberry. You're all drooling now, aren't you? So what I have to do is I have to go out and I have to harvest some strawberries. Wild strawberries are pretty tiny. Most of them don't really get much bigger than about dime-sized, which is really disappointing. And if you get them when they're really ripe, they're still somewhat tart, but some are really sweet and some aren't. So you'd want to take the sweetest strawberries, the ones with the best flavor, harvest some seeds off of those strawberries, and plant those seeds the next year. Hopefully, you'd end up with a bunch of sweet strawberries. And then repeat as necessary, plant the seeds from the best tasting crop that you got, and then the seeds from that next best tasting crop, until you end up with a population, a whole group of strawberries, your whole garden of strawberries that are just exactly what you want. And this is exactly where Darwin began his arguments too. Not necessarily with strawberries, but by looking at models of how humans have actually been selectively breeding animals over generations to try to achieve certain results. So artificial selection is one where the trait is one that's desired by humans. In natural selection, there's a bit of a difference. In nature, the trait has to improve your fitness, your survival or reproductive success. In artificial selection, humans deliberately choose which organisms reproduce, and in nature, this is something that is usually environmentally determined. Number 1, dead things do not reproduce. Zombies do not get together, do the wild thing, and produce baby zombies. This is not how zombies are generated. Dead things do not reproduce. Keep that in mind. You have to survive first, then you worry about reproductive success. So if your genetics don't allow you to survive to reproductive age, you're still not considered a very fit organism. Artificial selection is usually also a more accelerated model because we can really focus on what's breeding with what in the terms of animals, or even with plants. We can actually take pollen and move it around from plant to plant. We can very exquisitely control reproduction. In nature, it's a lot messier than that. Things happen usually more slowly, it usually takes longer, and there's one other big key difference here. Artificial selection can actually produce organisms that would not survive in nature. Take a Chihuahua. If you have to put your dog in a sweater, its survival chances in Alaska are very, very poor. So we can actually breed for traits or conditions or genes that don't appear to actually improve survival or reproductive value, except that we find them valuable enough that we will take extra nice care of those organisms, like your Chihuahua, and put sweaters on them, or whatever it may be. We do this for plants. When's the last time you saw banana seeds? You don't. Modern bananas are almost all clones. So we have to be very careful with how we handle those plants. We'll come back to some of those issues. Looking at antibiotic resistance and drug-resistant bacteria. This is a problem that affects a lot of people. To be honest, I have known people who have come down with MRSA. They're usually on antibiotics for several weeks to several months trying to clear those infections. And it's actually a fairly complicated set of problems. It's not just one thing. It's not just that they're penicillin resistant. They're resistant to penicillin, they're resistant to vancomycin, erythromycin, doxorubicin. I mean, they're resistant to almost everything we can throw at these bacteria, and they just don't die anymore. So why? Interestingly, this problem isn't new. It started back in the 1940s. Ironically, that's around the same time that we introduced penicillin. Typically, what I hear as the cause of this is the overuse of antibiotics. And you'll see this all over those Facebook things and all over the internet. But what do you mean by overuse? Does it mean that you took too much penicillin? Does it mean that you took it when you didn't need it? Sometimes yes to all of that. The thing that you need to keep in mind, antibiotics kill bacteria. They are useless against viruses and fungi. So if you have the flu, you don't need antibiotics. It's a virus. Antibiotics work on bacteria. They're not the same thing. Apples and oranges. And yet we still have people who go to the doctor who have the flu and expect the doctor to give them prescriptions for antibiotics. There's another part of this problem, and that's that bacteria reproduce really, really fast. Some of them in as little as 20 minutes. I've seen different numbers for this, but I'm going with my own personal observations. My first year as a graduate student, my project was to watch the growth and division of Pseudomonas aeruginosa in real time under the microscope. That's a bacterial strain that causes eye infections, particularly for people who wear contacts. It's also problematic for people with cystic fibrosis. These bacteria could split and reproduce in about 20 minutes. Sometimes a little more, sometimes a little less, but generally it took about 20 minutes. That's fast. So in the time it takes you to go through this whole chapter of information, your bacteria will have doubled at least 3 times. So if you started with 1 bacteria, you had 2, and then you had 4, and maybe even by the end of this material, you might now have 8. Oh, yeah. Kind of makes you want to dip yourself in bleach. So what we're actually looking at is the time frame between when we introduced penicillin and other antibiotics and when we first started seeing antibiotic resistance. So for penicillin, which was introduced in the early 1940s, to the time we first started seeing resistance was only 2 or 3 years. For chloramphenicol, it was even shorter. Erythromycin, a little bit longer, but still less than a decade, way less than a decade. Methicillin, less than a decade. Cephalothin, one of the first generation cephalosporin drugs, also way less than a decade. Most of these occurred within 2 or 3 years of when we first started using the antibiotic. That's a very, very grim statistic. So here's what you have to keep in mind. Up from about 1940, when we introduced penicillin, until the early to mid 1960s, you didn't actually need a prescription for any of these medications. In fact, you didn't even necessarily have to see a doctor. You could go to your pharmacist, say, my kid's sick, I need some penicillin. They'd hand you a bottle and you took it home. It didn't come with dosage instructions. It didn't tell you how much of it to give to the kid. It didn't tell you how many pills to give to the kid, how many times a day to give it to the kid, or for how many days to continue. It was up to your judgment. So people used it until they started feeling better. And they used it for everything. It was a wonder drug because it cured so many infections and it saved so many lives. And we used it, and even if we didn't know what the cause was, the general accepted practice was take some penicillin anyway. This is what led to the problem. So, what happens when you take an antibiotic? What you're looking at here is Campylobacter, and these are bacteria that typically cause food poisoning. They're one of the mechanisms of food poisoning usually found in contaminated meat. Animals do carry these, but they're usually asymptomatic. So these are gut flora, normal gut flora bacteria. And in 1987, we introduced a drug that interferes with bacterial DNA synthesis. It keeps the bacteria from copying their DNA and thus themselves, but without impairing our own ability to do that. Genius. So in nature, just out there in the wild even, some bacteria already have resistance genes and others do not. If you're thinking about penicillin for a starting point, penicillin is a fungus. It grows in the wild. It grows in the soil. It grows on bread. You don't have to have a pill for it necessarily for it not to exist. So there are probably already reasonably some bacteria out there that are drug resistant. Those are the ones that you see in red. The ones that are shown up in yellow, those are the ones that are antibiotic sensitive. And this is of our Campylobacter. So when we start administering the ciprofloxacin, the antibiotic, what happens is some of the bacteria die. Typically, those are the ones that were sensitive. The ones with the resistance genes, they tend to survive. A few of them may die, but not nearly as many. So over time, what happens is the drug kills off your sensitive strains and leaves behind your resistant ones. And this is exactly what happens. Your drug-resistant strains, especially your multi-drug resistant strains, become more common in your population. And that's a very careful way that you need to phrase those, especially in things like your essay question answers. They become more common, not dominant. Dominant is a genetics thing. We'll deal with that later. For now, becomes common or becomes rare. Try to keep it that way. So it's not just MRSA. We have other versions of antibiotic-resistant bacteria, including Pseudomonas, including Campylobacter, including Streptococcus, including MRSA. Hospitals today are major sources of infections. You might ask yourself, why? Stop and think about it. Who's in hospitals? Why do you go to the hospital? You don't. Good. Excellent. Try to avoid that if at all possible. But most people who are going to the hospital are going there because they're either sick or because they're injured. So these are problems, and the bacteria that we see in hospitals are exposed to more antibiotics because they're constantly in use, they're everywhere, and it's a perfect breeding ground. You've got sick people all over. So we're trying desperately to kill bacteria, and we have developed lots of antibacterial hand soaps and antibacterial wipes and antibacterial hand gels and all these other wonderful antibacterial products because we have decided that we are deeply afraid of bacteria today. There's a problem with that. Your hand sanitizer, guess what it's doing? It's actually contributing to breeding hand sanitizer resistant bacteria. I kid you not. Your antibacterial soap, same thing. Most of the antibiotic use is actually in livestock. In fact, something like 80% of all antibiotics are given to livestock, animals that we raise for food purposes. I had a student in my class a couple years ago who actually said this in class, that as soon as the pig is born, the first thing they do is give it a big old shot full of antibiotics. It hasn't even been out of the mommy pig for like more than a minute yet. First thing that happens, big load of antibiotics. Why? Why not? You want to ensure the health of your stock, of your animals, and this reduces the risk that they're going to be born and immediately exposed to infections. The problem is that, again, you're creating a breeding ground for these drug-resistant bacteria. In fact, today there is very, very, very little research going on into developing new antibiotics for us to use. It's becoming a major concern for human health. So what can you do? Things that bacteria cannot become resistant to, bleach. Bleach kills pretty much everything. It's still a pretty good way to go. Alcohol. Rubbing alcohol, like isopropyl alcohol, 70% or greater. Anything below 70% is not considered effective. That is the level that we require for lab use, so 70% or higher. Fire. Fire is pretty good at destroying most things too, although I don't recommend using it to sterilize your hands. It would hurt. The autoclave. This is a very high temperature, high pressure chamber, kind of like a pressure cooker that you actually put things in like glassware and liquids and waste, and the temperature and the pressure get so high that basically everything dies. Again, you don't see those typically for home use, but they are fairly common in barber shops, hair salons, nail salons, laboratory use. Filters, if you're interested in filtering out bacteria, you need one that is 0.5 micrometers or smaller in diameter. In fact, if you really want to get picky, it needs to be 0.22 micrometers or smaller in diameter to keep out bacteria. That's a really, really, really strict filter. Regular old hand soap also, oddly, works pretty well. The way soap works is that it breaks up the connections, the attraction between dirt and bacteria and your skin. And then the bacteria and the soap rinse down the sink. You don't care what happens to them. You don't have to kill them. You just want to get them off you. The thing is, though, that you have to wash your hands for 20 seconds. They've actually done studies, and if you go to med school or microbiology classes, you'll probably test this too. The studies show that if you wash for like 15 seconds, it's not as effective. And you don't get good optimal removal of bacteria until you hit about that 20-second mark. Could you get away with 18? Yeah, but you're getting kind of nitpicky here. So the other thing that you can do to help is apply the soap directly to your skin, especially if it's liquid soap, and get a nice lather and layer first, and then wet your hands with water, because that will surround all your dirt particles and stuff with the soap. And there's hydrophilic and hydrophobic interactions and chemistry that goes on in there, but you actually get better removal if you do it that way. If you don't believe me, try the next time you cook dinner. Conservation is another issue where evolution actually plays a role, and it becomes important. It helps to inform some of the decisions that we make about conservation. So what you're looking at is actually just a basic phylogenetic tree that shows the 3 major domains of life: bacteria in the blue, the archaea in the pink, and the eukaryotes in the yellow. Somewhere down here in the corner, we've got the metazoans, the complex multicellular life forms. That's us. We're a very tiny part of this tree. But what these are is really a branching representation of the connections between different living things. So if you're looking at the metazoans, the us's, you can see that the next closest related thing according to this is the choanoflagellates. Okay, whatever those are. Apparently they're single-celled, but other than that, I don't know much about them either. It's okay. You don't have to for this class. Our depictions of this are constantly shifting and changing as we get new evidence. This one is based on ribosomal RNA, the genetics of ribosomes. The things that are still hypothetical that we don't have evidence that agrees well for are represented with dashed lines. You'll see those fairly commonly. That just means we're not sure, or we have conflicting evidence and we're not quite positive this is the way it goes, but this is what we think might be going on, at least at this point. So, mammoths. Just 2 days ago, I actually read a thing on the internet that said mammoths may become a protected species. Wait, aren't they extinct? How did they go extinct? Why did they go extinct? Why would we have to make them a protected species? Are we planning to bring them back? I actually heard rumors about that several years ago, but I didn't realize anybody was going to try it. Did we not learn from Jurassic Park? Anyway, so if we had the choice, could we save the mammoths? Should we save the mammoths? This is an interesting question. Should we bring the mammoths back? I don't know. This is a choice that each person has to kind of make for themselves, and ideally you should want evidence and data and to have an idea what you're getting into. However, what we're looking at in this phylogenetic tree is the basic relationships between mammoths up there at the top and Asian elephants versus African elephants. And based on that tree, you can actually see that the mammoths are more closely related to Asian elephants, those branches connect, than they are to the African elephants, which are sort of down here in a little group of their own. That's where the genetics fall out, at least based on every bit of evidence we have to date. So what does this have to do with conservation? We couldn't really change what happened to the mammoths, but there are a lot of species today that we are very deeply concerned about. And I'm still a little worried about why mammoths might actually end up being a protected species. So this is actually fairly common throughout biology. Groups of things arise, species come about, and species go extinct. This is a normal process in nature, and it happens all the time. It's not all human hunting. We certainly did not hunt T-Rex into extinction. My version of what happens to T-Rex is I see T-Rex, I run the other way. Must run faster. I've seen Jurassic Park. Yes, and Jurassic World. But it's not just the dinosaurs. There's the mammoths. There are a lot of species that we worry about today, like cheetahs and whether they're likely to go extinct. We've had to do a lot of conservation work on birds, like some of the ones that you see here. Macaws and things, these become endangered species where we have very few of them left, and they're not really making a good breeding population. Like I said, it's not just due to human hunting or even human activities. Some of it may be. But there's also other options like drought, starvation, ice ages. You guys all saw the movie Ice Age, right? If not, you need to. Watch Ice Age because it's funny and I like Pixar movies. But it brings up a good point. Freezing to death is an equally valid mechanism of dying. And it's not one that you can control or that you can change or that you can necessarily do anything about. If an ice age happens, it happens. You either survive it or you don't. But one of our concerns is the impact that we as humans are having on our environment, especially as we pave paradise to put up a parking lot, to borrow a song lyric. As we modify our environments, as we tear down forests, as we disrupt ecosystems to build ourselves habitats and to build ourselves places of work, and as we mine ores from the ground to try to make new and better chemicals, and as we pump oil out of the ground, what impact does this have on the forms of life that are around us? We do actually have concerns about this. Some people do really worry about this. They tend to become conservation biologists. It's a really cool field. But it leads to some really difficult decisions. How do you deal with this? How do you make these decisions? Do you want to focus on saving the cheetahs? Do you want to focus on trying to save the kinglet calyptera down there at the bottom, or do you want to focus on the macaw in the pretty blue? Is the cherry-throated tanager more your speed? What do you choose? How do you save them? Because you can't save them all. And that's a very real problem. So we've got kind of two ways of looking at this today. One of them is to preserve phylogenetic diversity, or how branchy the tree of life is, how bushy it is. And the other one is to try to preserve as much of evolutionary history as we can. So in terms of looking at phylogenetic history, you have a couple options here. And I'm going to give you a minute to think about it. But I want you to look at this figure for a second and figure out what hurts the least. If you lose the group B and C shown there in blue and highlighted in blue, which is 2 species, or if you lose E, F, and I, which are shown in red and highlighted in red. Which is worse? Which would you have to pick one or the other. One of those 2 groups, blue or red, have to go extinct. Which one do you choose? Some of you want to say BC because it's only 2 species. Some of you want to go E, F, and I because although it's more species, if you lose E, you still have D. If you lose F, you still have G. Similarly, if you lose I, you've still got J, and even fairly closely related H. Both of these methods of looking at it are kind of valid. So, in this particular case, if you focus on saving this group, the BC group, you're focusing on preserving phylogenetic history. Loss of this group represents the loss of an entire branch off the tree, and a fairly large one and a fairly old one because this is a nice long line. Losing E, F, and I are relatively minor losses in some ways, if you're looking at it from the perspective of phylogenetic history. But you can also look at it from the perspective of phylogenetic diversity. So look at all the branching that goes on from this point forward in each of these 2 lineages. BC only splits once, but this other branch over on the other side splits into D, E, F, G, H, I, and J. That's a lot more splits. So sometimes when new species come about very, very quickly, this is called an adaptive radiation or speciation, and they can go hand in hand. So this is a rapid burst of new species forming for reasons that aren't necessarily clear, but does that make it more interesting? I mean, are these the future species? Should we be focusing our conservation efforts on saving these new things that are coming about, instead of the old things that are in the past, that are more ancient? How do you make these choices? This is one of the big things that affects conservation biologists. Understanding these relationships helps us to see some of these patterns, and so we may be able to make an informed decision like, it's going to hurt, but maybe we should let F go and focus our efforts and our money on trying to conserve C. Or maybe it's the other way around. You can argue it both ways. But if you're trying to preserve phylogenetic diversity and keeping the tree of life bushy and full and fluffy and frizzy and whatever terms you want to use, then you would want to focus your efforts on the red group. So in this last little bit of the chapter, we're going to take a look at empirical models for evolution and theoretical models for evolution. This sounds really complicated, but it's not that bad. Empirical studies are ones that are usually based on what you typically think of as observation or manipulation lab-type experiments, even if it's going out and just watching monkeys pick lice off each other. The theoretical models are more computer-based. These are usually mathematical models, and they have some really good points. They can help us understand how complicated systems work. So to give you a really good example, the 2016 Rio Olympics, this was during the height of the concerns about the Zika virus outbreak. And one of the things that was really interesting was that computer scientists on the theoretical mathematical model side predicted that there would be actually pretty low impact from Zika. The people in the biology fields were busy going, ah, and freaking out and going, no, we don't think so. Those computer people have got to be wrong. So at the end of the Olympics, how many reported cases of Zika did we find? 0. At least currently. And that's as of right after the end of the Olympics from 2016. Very, very few cases. So those mathematical models can really be helpful for us to understand like how diseases spread through populations. They can help us focus on how complicated systems work, and they can help us to find sometimes a little detail, one little gene that maybe causes a bunch of different effects, or the original source of an outbreak of a virus. And this can help us define our expectations, and it can help us do better science. We generally try to back up our mathematical models with actual, like, empirical studies dealing with observations or manipulations. So for just a minute, we are going to actually spend a couple minutes now looking at the ancestry of humans and a little bit about some of the evidence involved in this. Not in great detail. Again, we've got a chapter on that coming up in the future. But we have this lovely issue about ancestors and relatives, and I need to make a very clear distinction here, because there is a difference. I have ancestors. Those are the people in my direct lineage, my mother, my grandmother, my great-grandmother, my great-great-grandmother, but it doesn't necessarily include all my relatives, my third cousin 8 times removed. I don't even know who that would be. I don't even think I have one. So humans and chimps are relatives. We share a common ancestor somewhere way, 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 way eons back in time, but humans did not evolve from chimps any more than chimps evolved from humans. We share an ancestor, but I didn't descend from my cousin. Humans didn't descend from chimps. We'll look at that part again too. But just for now, keep in mind that the two words, although we often use them sort of interchangeably in our loose, everyday, normal language, they actually do have distinct meanings, and that's something to keep in mind. So, things that we actually can look at. We do have fossil evidence that shows a lot of anatomical similarities. We're going to look at some of that in just a minute. Molecular and genetic evidence, and there's even behavioral evidence. Your textbook actually doesn't even go into this very much, but we actually see evidence of altruistic behavior in chimpanzees. They will help each other. even if it comes with no benefit to them. We see evidence for parenting, and we see evidence for adopting baby animals of other species. So these are things that we connect with as being very human-like. And in fact, many people, if they go and they observe chimps or other primates in a zoo setting, they come away feeling vaguely uncomfortable because they are so human-like. So starting off with even stuff that Darwin observed. These are skeletons of gibbons, orangutans, chimps, gorillas, and humans. And you'll notice that although there are some striking differences, like the skull shape, there are also some rather surprising similarities, like the arm structure. As you look at it, you'll notice that they all have one upper arm bone, the humerus. There are two bones in the lower arm, the ulna and the radius. There's a bunch of little bones in here. You've got the carpals and the metacarpals and the phalanges that form the fingers, and your legs actually show the same structure, and the rib cages are kind of similar. The gorilla's is a little bit large and kind of more round. But our skeletons show some surprising degrees of similarity, and a lot of scientists and a lot of people look at this and think, that can't be coincidence. Why would these things all be structured the same way? What is the point of this little thing sticking out down here, the coccyx, the tailbone? It does not exist just so you can break it. Why do our skulls have the shape that they are? Why do you see these giant jaws in some of the others? These are things that we'll come back to when we get to the chapter on human evolution. But for now, this is where some of our earliest ideas on human evolution and the possibility that we have changed or that we shared a common ancestor with other primates, this is the kind of thing that starts raising questions for people. This is where we start wondering, is there a connection? Today, we actually look for chromosomal evidence, and we can find some chromosomal evidence. So what you're actually looking at are all of the chromosomes for all four species. You've got human, chimpanzee, gorilla, and orangutan there, A, B, C, and D. And the chromosomes are lined up, human, chimpanzee, gorilla, and orangutan. Where's the difference? Which set of chromosomes is not like the others? And I will give you some time to look at those and to study those and try to figure it out. See if you can spot the difference. I promise not to hum the Jeopardy theme song. Did you find it? Hopefully you did. Chromosome number 2 is different. Humans have one chromosome there, but chimps, gorillas, and orangutans, theirs look broken, and it's actually because they have two separate chromosomes where we only have one. Now you may be wondering why this happened and how we know that these are supposed to be the same chromosomes. Look really carefully at the banding patterns of light and dark, especially in these lower regions of the chromosome. I think it's most evident for you there. You'll notice how those spots line up on all of those chromosomes. That means they're all part of the same chromosome. And you actually see the same banding patterns that match up on the top parts of the chromosomes as well. When we did deeper gene sequencing and we actually looked at the DNA sequence, the ATGC stuff, we found out that sure enough, our chromosomes are actually a fusion of the two chromosomes that we see in all the other primate species. So we're the oddballs. Oops, we're the mutants. Ah! I want my X-Man powers. When they looked at the gene sequencing, they also found some other stuff. They looked at a group of humans and a small group of chimpanzees. This was not a huge study, but it's become really commonly cited. So they found basically about a 1.3% divergence overall between human genes and chimpanzee genes. But it's not consistent. It's not even consistent on the same chromosomes. Some of them had a lot of differences, some of them only had a few differences. They had to account for neutral mutations, which is a thing that we'll talk about later this semester too. And they found some really interesting differences in some different groups of genes. So for example, malaria resistance and tuberculosis resistance genes were unique to humans. We didn't even find those in chimpanzees, and they're our next most closely related species, according to DNA. We found other differences involved in sperm production and pregnancy protein production, so there's something different about human versus primate mating, chimpanzee mating. Good. Yeah, okay. There's clear differences in that. If you think about it, humans are actually one of very few species that doesn't have a classical estrus cycle. We don't go into heat. We are pretty much always receptive to sexual mating, unless the women have headaches. Although that happens for men too. Jokes aside though, we do have a very different mating strategy, and we are also different in terms of how our bodies react. A lot of animals don't have a menstrual cycle where they bleed every month either. So that is something that is different in terms of how we deal with this. The differences in sperm production are also a little bit intriguing. How and why that would be different for chimpanzees versus humans, you can get into the reproductive biology of that if it intrigues you. Even though a lot of the DNA wasn't all that different, in fact, it was like 98.7% the same, our genes aren't used the same way. Our genes that say grow hair here or make a toenail there, they don't necessarily turn on and off at the same times. They're not necessarily used in the same way. And that's something we'll talk about when we get a little bit further on in the semester too. If you look at the sex chromosomes X and Y in the bar graph on the left-hand side here, the X chromosome, you'll notice there were some differences that varied in some regions of this X chromosome between, that's 0.8%, very, very similar, up to 1.2% different. The Y chromosome showed a lot of differences between humans and chimpanzees, 1.6% different on the Y chromosome. Again, since we just heard that there were differences in sperm production, having that on the Y chromosome, that's a male thing, that actually makes some sense. When you look at the non-X and Y chromosomes, you see that there's actually a big spread in the data, everywhere from 0.8%, less than 1%, up to around 2% difference, 0.02. And that varies a little bit for each chromosome that you look at. So some vary more than others, some vary less than others. There's a fair spread. And most of them, it looks like, on average, work out to about 1.3% difference. So interestingly, they also found some other differences. Gene expression in the heart, liver, testes, and brain cells was pretty different between humans and chimpanzees. Although they figured that the heart and the kidney and the liver, that's similar to computer models that we made, so that's probably just noise. It's probably random chance that those genes are expressed a little differently. But that Y chromosome, that male mating thing, and sperm production, the testes were statistically significant. So this again goes back to the idea that maybe it's because we have very different mating systems. Maybe there's something biologically different enough about how males and females copulate in chimpanzees than in humans that they actually have slightly different genetics controlling that. What's really bizarre and slightly concerning is that they didn't see a big difference in brains. And we're supposed to be these smart people, you know, we're like the top of the evolutionary food chain and stuff, right? Yeah. There were some differences in gene expression in the brain, and there were some genetic differences, but not as much as we would have thought. But if you compare primates as a group to all the rest of the mammals, cats, dogs, mice, bunny rabbits, etc., there were big differences between humans and primates in general versus the squirrels. We are way smarter than a squirrel, thank goodness. Genetics in the brain isn't necessarily a measure of intelligence either, but it helps to kind of keep things in that perspective so that it's understandable. So where does that leave us? What does that make us? right now, humans are the closest living relatives of the chimpanzees and the bonobos, or bonobos. I've heard it pronounced both ways. I don't really care which way you pronounce it. You'll notice, though, that these two are actually grouped together. They are actually more closely related to each other than they are to us. That group diverged. It split after humans had already branched off. It's more recent. So this is a new thing going on, or a relatively new thing. But our understanding of this, our knowledge of this, goes all the way back to Darwin and Huxley and others who all noticed those early anatomical similarities in the skeletons. It's not new. And we're still arguing about it to this day. Modern techniques place more emphasis on genetics, so we tend to rely on DNA evidence more than we do on anatomical evidence because it's easier to count and measure. You get A, T, G, and C. You're not going to sit there and argue about, well, I think it's a T, I think it's a G. Okay, it's either a T or a G. Chemically, we can distinguish them very easily. So we can compare those sequences, and there's much, much lower risk of errors of interpretation. Last question for chapter 1. Finally, we get here. Why is there an even sex ratio? What's with this 50-50 male and female thing? And what does that have to do with empirical and theoretical models of stuff, of science? We're going to take a theoretical approach first, and then we're going to see if we can find an example in nature. So, in humans and in a lot of species, females are XX, males are XY. Hopefully you guys are good with that. You should have gotten that in an intro biology class. When males go to produce sperm, they produce half of their sperm carry the X chromosome and the other half of their sperm carry the Y chromosome because of the way the genetics divide up during meiosis. They basically just split, so half and half. Some of them are X, some of them are Y. Females, we do the same thing. The only difference is we have our X's, so yeah. This becomes important because it means that for humans and for most animals, it's the male that's actually responsible for chromosomal sex determination. They're the ones who determine the gender of the baby. It has nothing to do with what you eat. It has nothing to do with what position you have sex in. It has nothing to do with whether or not, I don't know, you stood on your head and twirled and prayed. It all has to do with whether you get an X or a Y carrying sperm. Because females, all we can do is say, female. So it's entirely up to the males. Somebody should have explained that to Henry VIII. The man had 6 wives, mostly because he wanted to get a male heir to carry on and inherit the throne. His first wife he divorced. He actually invented divorce to get rid of her. Second wife he beheaded. I guess divorce was too expensive. Third wife died in childbirth. Fourth wife divorced again. Fifth wife beheaded again. Sixth wife finally outlived him. Yikes. All to get a male heir. Yeah, and it was his bad. So, let's suppose that male births, whether human, cat, dog, or whatever, are less common than females. That means males are rare. A newborn male that comes into this situation is therefore very much in demand. They're the rare ones. Everybody wants to date them. They have their pick of all of the females. So they have better mating opportunities than a newborn female. The males don't have to compete with each other as much for access to mates. The females are going to be fighting over them instead. So this is a good situation for those males. Therefore, because they probably have better mating opportunities, in this case, the males are almost guaranteed to be able to mate. The females not necessarily. The males should leave more offspring. You with me so far? Hope so. Okay. Anything that you may have, any little genetic quirk that may run in your family that makes you more likely to have males means your genetics are therefore more fit. You are awesome. Your genes are going to get passed along more frequently because you produce more males. So those genes should spread through the population and they should gradually become more common in the population until one thing happens. Until you hit a 1-to-1 ratio, an even number of males and females, 50-50. What happens if you go past that? Well, now you have more males than females, so females are the ones that are rare. They've now got the benefit. It switches. So either way, whichever gender starts off as being rare in your population has an advantage in terms of mating because it's in demand. So for a real-world example of this, we have these blue moon butterflies that we actually see in various islands, and in 2001 they surveyed these butterflies and they found that 99% of them were female. The males were very, very rare. They only constituted about 1% of the population. So this is exactly what we just talked about. This was actually surprisingly due to a bacterial infection. The males were more susceptible. They died, but the females survived. Oddly enough, I've seen this in my own research with mice too. The mice, when I was dealing with the horrible brain infections, actually we had to give them a higher dose of virus to get them to the same level of sickness that we gave the males. So there's something about being female that seems to give us a little bit of an immune advantage. That disappears, by the way, as you get older. So what should happen based on our previous understanding? Males are rare. They've got an advantage. Any gene that makes male is an awesome gene. So if some of the males are naturally resistant to the bacteria, if some of the males just throw off lots of Y chromosomes and produce lots of male offspring, these are beneficial traits. These are useful characteristics to have. And those genes should spread through your population. Your males should start to recover and should get to about 50-50. Any genes for resistance? Those are going to be the cause of that. So they should also increase. So just 5 years later, in 2006, they went back and they looked at these blue moon butterflies again. And they found that in many of the areas they'd actually seen good recovery of male blue moon butterflies. In Salelua, over here, down here on this coast, 46.3% male, not quite at 50-50 yet. Apia, 49.3%, very close. Saleluga, 58.3% male. Whoops, they overshot a little bit. And then over here you've got Sagone, which is only 7% male. Does that make you wonder why? If all the other butterfly populations have rebounded, what's wrong with this group? Maybe they don't have as many resistance genes. Maybe there's something else going on in that region. Maybe it's starting, but it just hasn't gotten there yet. I mean, it's better than 1%, so that is some recovery. They went from 1% to almost 50%, or in some cases even more than 50% in 5 years. This is genetic change over time. This is evidence for evolution. It shows us that the genetics, the percentages, the numbers of things that we can go out and count really do change, sometimes in startling and measurable ways, even in a surprisingly short time span. This is just 5 years, but it shows us what's possible. So this is where things begin, and this is the end of Chapter 1.